Evaluation of hemoglobinopathies by HPLC, prepared by Marco Flamini for Viral Laboratories. This presentation is focused on hemoglobins, hemoglobin disorders, and hemoglobinopathy testing by HPLC. We will review the synthesis of hemoglobin during the different stages of human development, basics of HPLC separation, the relationship between globin genes, their expression, their genetic alterations, and the evaluation of phenotypes through the review of chromatograms. Hemoglobin is a complex protein, a big molecule with a tetrameric structure made of two dissimilar pairs of globin chains, alpha or alpha-like and non-alpha or beta-like. There are 4 heme groups with iron atoms to bind oxygen. Transport of the oxygen from lungs to tissues is the main function of hemoglobin. Hemoglobin molecules fill the erythrocytes, red blood cells, giving to blood the typical red color. There are almost 300 million hemoglobin molecules inside each red blood cell, and in average there are 5 million red blood cells per microliter. Around 14 grams of hemoglobin per deciliter, and each red blood cell contains 30 picograms of hemoglobin, which represents the mean corpuscular hemoglobin, MCH, in an 85 femtoliters of volume, which represents the mean corpuscular volume, MCV. We will see that depending on the amount of available hemoglobin, the volume, MCV, and the quantity, MCH, per each red blood cell could be reduced, as in anemia. Hemoglobin in adults is produced in the bone marrow. Red cells have an average lifespan of 120 days and finally are destroyed primarily in the spleen. The gene clusters for hemoglobin chains are on chromosome 16 for the alpha and alpha-like chains, and chromosome 11 for the non-alpha chains. Genetic alterations may involve the genes, but also the loci control regions and the enhancers. DNA encodes the information for making globin chains. The sequence of bases is transcribed into RNA and translated to make a sequence of amino acids, resulting in polypeptide chains, which are the globin chains. Alpha chains are constituted of 141 amino acids and bind with non-alpha chains that have 146 amino acids. The non-alpha chains are beta, gamma, and delta chains. We inherit chromosomes from father and mother, and we have then 4 alpha genes, 2 beta genes, 2 delta, and 4 gamma, encoding for gamma chain of hemoglobin F. Just a note to show that commonly the alpha-2 gene is responsible for synthesis of more alpha chains than the alpha-1 gene. The quantity of alpha and non-alpha chains are equal in normal conditions. Two alpha chains bind two beta chains to make hemoglobin A, the adult hemoglobin. Two alpha chains bind two gamma chains to make hemoglobin F, common in newborns. And two alpha chains bind two delta chains to make hemoglobin A2, which is present in adults at very low levels. This graph shows the production of the various chains at different stages of human development: embryo, fetus, newborn, infant, and adult. Blood and hemoglobin is considered adult after 1 year, when the physiological switch between hemoglobin F and hemoglobin A is completed. The 4 alpha genes encode the alpha chains that are expressed during the whole period, from fetal through adult. In the fetal stage, the main non-alpha chains are the gamma chains. Beta chains synthesis start just before birth. Delta chains are not present in newborns or in very low quantities. During the first 6 months of life, there is a switch in the expression of the genes from gamma to beta. There is a change in the hemoglobin pattern from hemoglobin F to hemoglobin A that can be seen if the infant is tested at short intervals. And here is an example where we see how the level of hemoglobin F, alpha-2 gamma-2, drops down significantly in the first 3-4 months of life, counterbalanced obviously by an equivalent increase of hemoglobin A. Please note that hemoglobin F is slightly higher in beta thalassemia carriers, which means hemoglobin A will be lower in beta thalassemia carriers. And in fact, a reduction of hemoglobin A in newborns is studied to predict beta thalassemia. Gestational age, though, should always be kept in consideration. Only after 3-6 months we can see an increase in the delta chains that will contribute to making hemoglobin A2, alpha-2, delta-2. This is the making of hemoglobin A2 after birth. There are only traces of A2 at birth, and then we see the increase in A2 in the first year of life in normal and beta thalassemia carriers. A difference can be seen starting from the 3rd or 4th month, and it will be complete only around the 9th month of life. So there is often no hemoglobin A2 in newborns. These are the genes expressing globin chains in newborns: alpha, gamma, and beta inherited from our parents to make the main fractions in a newborn: alpha-2 gamma-2 hemoglobin F, and alpha 2, beta 2, hemoglobin A. A typical pattern in a newborn is with 80% hemoglobin F and 20% hemoglobin A. The ratio between alpha and non-alpha in a normal subject is equal to 1. In the adult, the switch has happened. 2 beta genes are in full expression. The gamma genes have largely stopped their production, and also the 2 delta genes are expressing. The main hemoglobin fractions in adults are hemoglobin A, hemoglobin A2, and a little bit of hemoglobin F. The ratio alpha-non-alpha is also here equal to 1. Let's talk about the charge of the hemoglobins. Amino acids can be polarized depending on the environment they are in. A different sequence of amino acids make different polypeptide chains and gives a different charge to the hemoglobin fraction. Hemoglobins can be then differentiated based on that charge. Here is the basic scheme of an HPLC system. 2 buffers are used to create a continuous gradient of ionic strength when pushed by 2 pumps or pistons through the cartridge. The separation of the hemoglobin fractions based on their different charge happens in the cartridge. Once released from the cartridge, the hemoglobin fractions reach the detector at different times, where they are detected based on the absorbance of energy and integrated into chromatographic peaks. Each peak represents a fraction, and the area under the peak represents its percent of the total. The best resolution power in the separation of hemoglobins is obtained creating a fine-tuned competition between the positive charge of the hemoglobins and of the buffer salts, also called liquid phase, with the negative charge of the column resin or stationary phase. The liquid phase changes its composition during time to be more effective in separation power. Obviously, 2 pumps and 2 pistons are much better than 1 to create a fine gradient of ionic strength. Here is a newborn sample. Hemoglobin F has a weaker positive charge than hemoglobin A, and it's eluted before it. 80% of F and 20% of A is common in newborns. Here, a chromatogram of newborn blood with almost 90% of F eluted at the beginning of the run and some 11% of A immediately after. We have to highlight that hemoglobin F is partially acetylated, and the high power of HPLC is able to differentiate this fraction from hemoglobin F. When hemoglobin F is very high, 40% and higher, like in newborns, the F and F1 peaks fall in different windows, labile and A1B. Knowing it is a newborn blood, it's easy to assign those peaks to to F and F1, also because labile and A1B are never so high. A chromatogram of adult blood showing common fractions resolved based on their different charge, including A1C. High-resolution HPLC is able to detect also post-translational adducts like A1C or other minor fractions, which are present in all patients. Those little peaks before A0 and F can be important to unveil the possible presence of a variant coeluted with A0. And this is important if we consider that there are more than 1,000 hemoglobin variants. These translational adducts are important also because of degradation products or to suspect other conditions. Hemoglobinopathies are hemoglobin disorders due to alterations in the genes on chromosome 16 and or 11. These can be point mutations or deletions that can involve any gene or the regulatory regions of the genes. These alterations can be in the heterozygous state, meaning isolated to only one gene, maternal or paternal, alpha or beta, for instance, or in the homozygous state, affecting both genes, which is more severe from the clinical standpoint. If the mutations are different like these on the beta genes, it can also be called double heterozygote or compound heterozygote. The genetic transmission of common hemoglobinopathies is autosomal recessive for most of them. The carrier status is commonly a healthy condition, but there is a risk on every pregnancy to have 25% of the offspring affected by a homozygous or double heterozygous condition. Knowing the carrier status of the parents is of utmost importance for reproductive risk assessment. There are two main groups of hemoglobinopathies. Those where the genetic alteration is causing a qualitative change in the structure of the hemoglobin, and we will have then hemoglobin variants such as hemoglobin S, hemoglobin E, hemoglobin D, hemoglobin C, and those affecting the quantity of the hemoglobin called thalassemia, such as alpha or beta thalassemia. Co-inheritance of different defects is also common in hemoglobinopathies. This is an example of alpha thalassemia heterozygote. Alpha 0 thalassemia is a large deletion that affects both alpha genes on the same chromosome and totally loses the production of alpha chains from that chromosome. The other alpha genes on the other chromosome are working well, as well as the non-alpha genes, beta, delta, and gamma. There is an excess of beta chains and less total hemoglobin. RBC are then smaller, low MCV, and there is less hemoglobin in each one of them, low MCH. This is a double heterozygote condition on the alpha genes. Alpha plus on one chromosome and alpha 0 on the other. There is only one functioning alpha gene very low alpha chains are produced and an important excess of beta chains is seen. Total hemoglobin is much less. The RBC are very small and subject to hemolysis. This is Hemoglobin H disease. With only one functioning alpha gene, the excess of non-alpha is quite significant. And this subject will make a tetramer of gamma 4 at birth, hemoglobin Bart's, and a tetramer of beta 4, hemoglobin H, after the switch from gamma to beta. A newborn with hemoglobin H disease will have hemoglobin Bart's at birth and will become an adult with hemoglobin H. These are the chromatograms with Bart's as a newborn and the chromatogram with hemoglobin H at 1 year of age. A hemoglobin H chromatogram in adults is typical because of the double peaks at the beginning of the run and a very low hemoglobin A2, lower than 2%. Moreover, they have altered CBC indices and inclusion bodies after viral staining on microscopy. DNA testing is required for reproductive risk assessment. Thalassemia heterozygote, a healthy carrier, is commonly due to a beta mutation, which leads to reduced or absent production of beta chain messenger RNA by one of the two beta genes. The production of beta chains directed by that chromosome is thus diminished beta plus or absent beta 0. Due to this defect, the other non-alpha genes delta and gamma on the same chromosome, which are close to the affected beta gene, are stimulated to compensate such a deficiency in the beta gene. That is the reason why we will have high hemoglobin A2 and often slightly high hemoglobin F in beta thalassemia carriers. Only one beta gene is producing beta chains, and we will have in the newborn alpha 2 gamma 2 F and alpha 2 beta 2 A. The amount of hemoglobin A in a beta carrier newborn is commonly lower than in normal. In adult carriers for beta thalassemia, hemoglobin A2 is typically raised and often also hemoglobin F. The excess of unbound alpha chains causes damage to the RBC membrane. There is less hemoglobin, anemia, in only a minority of heterozygotes, whereas microcytic erythrocytes with a reduced hemoglobin content are a consistent feature. Low MCV, low MCH. This paradox is accounted for by the increased red cell count. In the homozygote status for beta thalassemia, both beta genes are affected by genetic alterations. In the most severe forms, the synthesis of beta chains is totally absent. Only hemoglobin F and hemoglobin A2 are formed. In fact, only alpha and gamma chains are synthesized in the newborn, who is apparently healthy, but after 6 months, the switch to beta, it doesn't happen, and so the patient is left without hemoglobin A. Some A2 is detected, but no hemoglobin A. The clinical signs will start because of the absence of beta chains and more importantly, because of the excess of alpha chains which are insoluble. Severe anemia is the main problem. In a chromatogram, only hemoglobin F and A2 is detected. F with the acetylated form F1 and A2. You can see the enormous excess of alpha chains and the little quantity of total hemoglobin. RBC have a very abnormal shape and there is a severe anemia. Patients will survive only with blood transfusion that is needed to supply hemoglobin A. Compound heterozygous conditions where one gene is totally non-functional and the other has a minor alteration, beta plus, which allow a minimal production of beta chains is called beta thalassemia intermedia. A low amount of A is present and the severity of the hematological disorder is less. Here is an example of a chromatogram of homozygosity with two beta plus mutations. Both affected genes are producing a low amount of beta chains. Together with high hemoglobin F and A2, there is some hemoglobin A, but always in reduced quantities, lower than 50%. This is a case with quite high hemoglobin A. Most likely this subject has a mild condition. Remember then, when considering thalassemia cases, chromatograms, we have to rule out blood transfusions because that amount of hemoglobin A could have been provided with a blood transfusion. We will have qualitative defects when a mutation is responsible for the production of a different chain, in this case a beta chain variant, like hemoglobin E. It's a beta hemoglobin variant which is produced at a slightly reduced rate and hence has the phenotype of a mild form of beta thalassemia. Hemoglobin E is an extremely common structural hemoglobin variant due to a single point mutation on the beta gene. At position 26, there is a change in the amino acid sequence from glutamic acid to lysine. As one beta gene is expressing normal beta chains and the other is expressing beta E chains, we will have the following globin chains pool and patterns. Hemoglobin F, hemoglobin A, and hemoglobin E in the newborn. Hemoglobin A, hemoglobin E, hemoglobin A2, and some hemoglobin F in the adult. The mutation at position 26 makes the beta E chain more positive than hemoglobin A. For this reason, on HPLC, E is eluted after A. This is a typical HPLC chromatogram of an E carrier. The main peak is hemoglobin A, and the other in the A2 window likely hemoglobin E, which is more than 30%. We know that hemoglobin A2 is never higher than 8–9%. So that peak of more than 30% is likely hemoglobin E co-eluted with hemoglobin A2. It is always important to remember that any variant peak on HPLC, but also on all the other first-level methods, is only presumptively identified. When hemoglobin A2 is separated from a presumptive hemoglobin E, we have the issue that A2 is elevated in the beta thalassemia range. This can raise diagnostic questions. Hemoglobin A2 in the presence of variants with any methods must be carefully evaluated because of possible interferences. Practically, hemoglobin A2 loses its diagnostic significance. It's the presence of the other peaks, like A and the variant, that explains the possible phenotype. In fact, if hemoglobin E is associated to beta thalassemia, the main change happens with the disappearance of hemoglobin A because there are no beta normal chains and a considerable increase of hemoglobin E from 30% to more than 60-70%. Moreover, the phenotype E beta thalassemia is often associated with an increased production of gamma chains, hence high hemoglobin F. This is a visual representation of the E beta 0 phenotype. There is no production of beta chains normal, because one beta gene is affected by beta 0 thalassemia, the other gene is producing beta E chains. We will have an FE pattern in the newborn, while in the adult we have high hemoglobin E, no hemoglobin A, high hemoglobin F, and an increased hemoglobin A2. The presence of hemoglobin E higher than 50%, no hemoglobin A, and 15-30% of hemoglobin F suggests E beta 0, regardless of the level of hemoglobin A2. There is anemia, low total hemoglobin, and low indices MCV and MCH because of the reduction of the beta chains. E beta plus is due to a combination of one beta gene affected by a beta plus mild mutation and the other by a beta E mutation. The beta plus gene will lead to production of a low quantity of normal beta chain that will combine with alpha to give low hemoglobin A. Beta E chains will make more than 50% of hemoglobin E. Some hemoglobin F can also be synthesized. Here is a chromatogram of E beta plus with a very low quantity of hemoglobin A, high hemoglobin E, and high hemoglobin F. In the case of homozygous E, both beta genes are producing beta E chains with no normal beta. It is similar to E beta 0, but in this case, as both beta genes are expressing, there is less imbalance between alpha and non-alpha. The amount of total hemoglobin is higher than in E beta 0, and the thalassemia phenotype is less severe. Hemoglobin F is rarely elevated. Hemoglobin A2 can be raised, but with no diagnostic relevance. On HPLC, we will have a very high peak of hemoglobin E, 80–90% in the A2 window, no hemoglobin A, some hemoglobin F. Hemoglobin E homozygosity is not a disease. RBC are slightly reduced. Separation of A2 doesn't add any meaningful information. The chromatogram instead has all required information. A correct interpretation of a chromatogram, assessed together with the main hematological features, is fundamental in the evaluation of the hemoglobin pattern. A normal sample shows a very high peak in the A window, more than 90%, and A2 normally lower than 3.5%. RBC indices in the CBC, complete blood count, are normal. MCV higher than 79 femtoliters, MCH higher than 27 picograms, and total hemoglobin higher than 13–15 grams per deciliter. From the chromatogram and from normal indices, we understand that the main peak is hemoglobin A, alpha 2, beta 2, and that both beta and alpha genes are properly expressed. In a beta thalassemia carrier, the key feature is represented by a raised hemoglobin A2, which is never higher than 9%. Typical is also the low MCV and low MCH. We understand that A2 is raised to compensate for one beta gene not working, while the other beta gene is working well to make hemoglobin A, alpha 2, beta 2, even if the total quantity of hemoglobin is reduced. In a hemoglobin E carrier chromatogram, we see two main peaks, one in the A window, 60–70%, and the other in A2 window, some 30%. We know that one can't be A2, but hemoglobin E coeluted with hemoglobin A2. We envision one normal beta gene to make hemoglobin A and one beta E gene to make hemoglobin E. Resolution of A2 from hemoglobin E doesn't add any further information, but only possible doubts, because A2, when separated, is always in the beta thalassemia range. It is easy, the interpretation of HPLC chromatograms, and from left to right, we have normal A2, elevated A2, and very high A2, which is clearly hemoglobin E. Homozygous E shows one single big peak of around 90% in A2 window, no hemoglobin A, and maybe a little of hemoglobin F. This is the result of both beta genes carrying a beta E mutation and expressing beta E chains. This condition is not a disease. CBC is only slightly affected. When beta thalassemia with a mild mutation is associated to hemoglobin E, we will have hemoglobin E peak in A2 window in the range of 60–70%, with little hemoglobin A between 15–20% and raised hemoglobin F, typical of the thalassemia conditions. In the E beta plus thalassemia, we explain one beta gene is leading the production of very few beta chains, and the other only beta E chains. There is an imbalance alpha-non-alpha, as in all beta thalassemia, which results in abnormal indices. In case of E beta 0 thalassemia, we have hemoglobin E peak in A2 window in the range of 60–70%, no hemoglobin A, and always raised hemoglobin F, 20–30%. We explain one beta gene is totally switched off by a thalassemia mutation, and the other is producing beta E chains. As one beta gene is totally inactive, there will be a significant imbalance alpha-non-alpha with precipitates of alpha chains that will contribute to low MCV, low MCH, and low hemoglobin. E beta 0 thalassemia is a severe disorder that generally requires blood transfusions. It is always important to have genetic confirmation because the severity of the clinical condition is based on the mutation type. HPLC chromatograms from left to right show significant differences looking at the very high HbE peaks, the absence or heavily reduced hemoglobin A, high levels of hemoglobin F, and meaningful differences in CBC data. This study shows how hemoglobin A2 is raised in the presence of hemoglobin E, and so doesn't have any diagnostic relevance as it may cause misinterpretations. This table confirms the different levels of hemoglobin E between E heterozygote, E homozygote, and E with beta thalassemia. Furthermore, the level of hemoglobin F is very different between HbE homozygote and E beta thalassemia, which helps in the differential diagnosis. Also, in this table, we can see which are the parameters that can help in the differential diagnosis of hemoglobin E homozygote, E beta plus, and E beta 0 thalassemia. We see the different percent and low quantities of hemoglobin A, the different levels of hemoglobin F, and it is of interest that hemoglobin A2 is summed up to hemoglobin E. On summary, we have reviewed how the understanding of the expression of globin chains helps in the interpretation of chromatograms, and how important is the accuracy of hemoglobin A2 determination in beta thalassemia testing. Remember that using first-level methods, the identification of hemoglobin variants is always presumptive, and that in the presence of hemoglobin E, A2 is raised and has different ranges from normal. Furthermore, we reviewed how important in differential diagnosis is the relative percent of hemoglobin A, hemoglobin E, and hemoglobin F values.